All right, this is just a little talk about muscles and muscle physiology. So, skeletal muscles, these muscles that are under voluntary control, contain many muscle fibers or muscle cells. So, many muscle cells make up one skeletal muscle within the body. And within your biceps, typically you're kind of predetermined with the number of muscle cells that you have. There's not really a wide variety in terms of how like changing the number of muscle cells within a muscle belly. You can modify it a little bit. There's some instances where you can increase the number of nuclei within a muscle cell, and there's some division that can potentially happen. But usually you're kind of set with the number of muscle cells. It's genetic, but you can increase the diameter or the cross-sectional area of a muscle cell. And what that means is you're adding more of these contractile units called myofibrils. So myofibrils are these contractile units where they're positioned, each one of these little white guys is considered a myofibril, and they're positioned longitudinally within this muscle cell. And each one of these myofibrils contains what are known as sarcomeres. So sarcomeres are individual contractile units. It's where I have a Z line on this side and a Z line on this side. And then I have these things called thin filaments, which are made up of actin. So these are thin filaments. These are thin filaments. And then I have these thick filaments, which contain myosin. And myosin, what happens is it'll come over and it'll bind to the thin filaments, the actin filaments, and it'll pull it one way on one side of the M line. And on the other side, it'll pull it the other way. So they're kind of moving in antagonistic directions. But overall, I'm shortening the size of this sarcomere. And if I do it, if there's many sarcomeres within a myofibril, the entire myofibril will change size. And if my muscle cell or my muscle fiber is made up of many myofibrils, the muscle will also change size. So the muscle is getting shorter, it's contracting when each one of these little sarcomeres is contracting that are within a myofibril. And so this is more of like the actual physiology or the movement of molecules that dictates whether or not a muscle will contract. And it's initiated by motor neurons that are coming out of the ventral horn of the spinal cord and that go and innervate muscle fibers. So a motor neuron can branch and it can innervate many different muscle fibers or muscle cells. And so this is an example of this motor neuron. Typically, it's triggered to fire within the spinal cord, and there's a wave of depolarization that goes down the axon of the cell, which typically has a relatively negative membrane potential. Now we're making it more positive. We're depolarizing the membrane by opening voltage-gated sodium channels. So imagine there's like a bunch of voltage-gated sodium channels on this membrane. They have a bottom gate that's typically open and a top gate that's closed. Bottom gate's open, top gate's closed. But when I get this wave of depolarization, the top gate will open momentarily. Momentarily the top gate opens. There's a brief window where sodium can rush from the outside to the inside of the cell, depolarizing the membrane even more, causing this next guy to open. But then the bottom gate will just immediately close. So I'm getting this wave of depolarization moving down the cell. And towards the bottom of the cell, I have what are called voltage-gated calcium channels. So these guys, voltage-gated calcium channels, are present on the axon terminal of this motor neuron that's going and innervating this muscle cell. And what happens is when I get this positive charge that's accumulating down here, it causes the opening of a voltage-gated calcium channel. So this guy is now opening. And calcium, which is actively pumped out of the cell, will move in following both a concentration gradient and a charge gradient. And it can bind to proteins that sit on these little vesicles on the axon terminal region of this motor neuron. And what happens is when calcium, imagine this little blue dot is calcium, binds to these proteins, it facilitates the movement of each of these vesicles towards the outer membrane. And what happens is the membrane of these vesicles right here will fuse with the outer membrane, and it'll release the neurotransmitters or these little signal molecules that are kept within these vesicles. And those signal molecules are called acetylcholine. And when acetylcholine is released into the synaptic space, there's an enzyme located on the surface of this muscle cell that immediately starts degrading acetylcholine. So I release a signal. Let's say this little signal is acetylcholine, but immediately I start breaking acetylcholine down into acetate, and choline. So I'm releasing a signal. It's immediately being broken down. But along with this enzyme called acetylcholine esterase that breaks down acetylcholine, I also have a channel right here positioned on the membrane. These are all channels called nicotinic receptors. And what nicotinic receptors are is they're ligand-gated sodium channels, and they're responsive to a ligand. And in this case, the ligand is acetylcholine. So when acetylcholine comes over and binds to these receptors, it causes them to open, allowing sodium to move from the synaptic space or the interstitial fluid into the cell. So sodium's rushing in. When sodium moves into the cell, sodium has a positive charge, so it begins depolarizing the membrane. And it depolarizes the membrane enough within this neuromuscular junction right here. This is the neuromuscular junction, to cause voltage-gated sodium channels to open on the membrane of this muscle cell. And voltage-gated sodium channels are positioned throughout the membrane, so a wave of positive charge is going along the entirety of this membrane, the entirety of the membrane of the muscle cell. When I reach the threshold or the charge that's accumulated on this membrane, that's enough to open the first voltage-gated sodium channel. And so muscle cells have this unique physiology where they have these invaginations of their membrane that go way down deep into the cell called T-tubules. So these are called T-tubules. And what it allows is it allows for this wave of positive charge to move down deep within the cell, where I have this channel right here called a DHP receptor. So DHP receptor, DHP channel, which in most tissues, like in the heart, a DHP channel is an L-type calcium channel, which allows calcium to move from the outside of the cell to the inside of the cell. But in this case, it's a protein that's detecting voltage, but it's not allowing calcium to move from the outside of the cell. So calcium out here is not moving in. So it's trying to, but it's not moving in. But what happens is when I get a positive charge that's moving down this T-tubule, it changes the shape of this DHP receptor. And this DHP receptor is mechanically gated or connected to this other channel called an RYR channel, called a ryanodin receptor. And what happens is when this DHP channel is changing shape, it changes the shape of this little connective tissue protein that's holding the DHP to the RYR, and it allows this RYR to open. And this RYR is a channel that's present on this space right here that's inside the cell but outside of the cytoplasm. So all of this space in here is a space that's unique within the cell because it's not cytoplasmic space. And it's a space where the cell actively pumps calcium into. So it's called the sarcoplasmic reticulum, where there's a bunch of calcium channels present on this membrane right here. And what happens is they're using ATP to actively pump calcium from the space within the cytoplasm into this space. So all these little green molecules right here, all these guys are supposed to indicate calcium ions. So there's a lot of calcium within this sarcoplasmic reticulum, upwards of 10,000 times the concentration of this space out here, the interstitial space. Because I'm pumping calcium so actively, I'm using ATP to move calcium against its normal concentration gradient and its charge gradient into this space. And so when I have this positive charge that's moving down the membrane, down this membrane, causing the change in shape of this DHP receptor, which is mechanically connected to this RYR receptor, it opens this RYR receptor and allows calcium that's been pumped into this space that's at a super high concentration to move following both a concentration gradient and a charge gradient. Because there's a lot of positive charge in here, the cytoplasmic space is a little bit more negative, so it's moving out following charge and concentration. And what happens is when calcium is released, so this is depicting calcium being released, calcium can come over and it can bind with a protein. So it comes over and it binds with this protein, this little yellow protein right here. This yellow protein is called troponin. And so troponin is a protein that's sensitive to calcium. It can bind with calcium. And when troponin binds with calcium, it changes the shape of troponin a little bit. So changing the conformational shape of this protein slightly, which changes the shape of this protein that's wound around this actin filament. And so when troponin changes its shape, it changes the shape of this protein right here, which is called tropomyosin. Tropomyosin. And if the shape of tropomyosin is changed, it shifts it just slightly out of the way. So it's just moving it slightly out of the way. This snake that's wrapped around actin is moving a little bit, which allows these myosin heads right here to bind with the actin filament. And when the myosin head binds with the actin filament, then you can initiate a muscle contraction. And it has to do, so this is the troponin molecule right here. This is troponin, this is calcium. This blue guy right here binding to troponin is calcium. This filament is tropomyosin. This is actin. This is the myosin head right here. And so when, I'm gonna go to this diagram. When calcium binds to troponin and changes the shape of tropomyosin and allows this myosin head right here, this myosin head, to bind with actin, which is this filament right here. When myosin is allowed to bind with actin, myosin typically holds ADP and an inorganic phosphate in its binding site. And what happens is the phosphate is ejected or pushed out. And remember, myosin is a walking protein, so it's facilitating the hydrolysis of ATP. It's not actually being phosphorylated in this process, but it kicks out this phosphate, and this causes the power stroke, which is this forceful movement of the actin chain by myosin. So myosin's pushing this way, it's moving actin this way. And then what happens is this ADP molecule that's within the active site of myosin is also ejected. So this guy is ejected from myosin. Now myosin needs ATP to be present in order to unbind from the actin filament. So you can see here, now that we have ATP around, and ATP floats over and enters the active site of myosin, it allows this myosin head to unbind from actin. And then this myosin head is a walking protein. So it's facilitating the hydrolysis of ATP, or it's facilitating the reaction where water will come over, H2O will come over and interact with the gamma phosphate on ATP, causing it to break off and form ADP and an inorganic phosphate. And then the cycle can repeat again in the presence of calcium.